Hello, everyone. I want to start out by thanking all of our viewers and welcome you all to Medical Student Grand Rounds. I'm Bryce Ringwald, fourth-year medical student and vice president of Student Council. Medical Student Grand Rounds is a lecture series given by inspiring medical students, and this series occurs on the first and third Tuesdays of the month, starting at 12 p.m. and is conducted via Zoom. Today, we have a fantastic presentation lined up for you all with one of our brilliant MSTP students, Brian Hansen, who will be presenting on his research project on cardiac arrhythmias and the future of rhythm mapping. And then on December 1st, we will be hosting our final webinar of the year. Shan Lansing will be presenting on the importance of rural healthcare and her work as a government advocate, working with representatives in Washington, D.C. Before we start, we are still in need of medical student speakers for our lecture series. And we ask that you use this QR code found here to nominate any potential speaker that you want to hear from. These can be self-nominations, or you can nominate someone else. We want to represent medical students across all classes who are doing amazing things and give them the opportunity to present and inspire their peers. During today's talk, we will be utilizing the Q&A function to gather questions during the presentation. We cannot guarantee that we will get to all the questions. We will do our best to get to as many as possible. It is my great pleasure now to introduce Dr. Vadim Fedorov for today's introduction.
Hello, everyone. I wish everyone stay healthy, strong, and fighting against COVID. So it's my big pleasure and honor to introduce, I would say, my best student, Brian Hansen. So Brian came to me more than 8 years ago as an undergraduate. He was just sophomore looking for research project and with primary interest go to med school. And when he came to me and we start working together, I saw great potential in this kid. We start doing very amazing experiments. And at this time, I was just assistant professor. So eventually, together with Brian, we published 30 papers. So Brian was just undergraduate student who came first time at Gordon Research Conference, one of the important conference which changed, I think it changed his life and introduced him a new perspective in his life, be also not only MD, but PhD, be medical scientist. So we work together day and night on the very exciting project, which he will present today. It's Ex-vivo human hearts. We have ability to study live human hearts with history of cardiac disease. We have great program developed here with direct help Brian. I wouldn't do it without him. With direct help of Brian, it's Ex-vivo human heart program. It's unique program across the world. We only the team in the Ohio State University who can do this integrative research, 3D imaging research of the human atria, and developing translational direct application to treat one of the most common cardiac arrhythmia, atrial fibrillation. Brian, during these years, developed exceptional skills and leadership skills. So I knew that during this long 24/7 experiment, I can always rely on the Brian. So he successfully presented this research in more than 40 different international and national societies, conferences. He received great awards across the field, including NIH F31 award. In 2018-19, he was nominated and received one of the most prestigious award at the Ohio State University, OSU Board of Trustee student recognitions. So he has great personality, so wonderful person, and I'm so happy that our scientific career for this many years, almost 8 years together, we were so successful. Here, I would like to introduce Brian Hansen and his very exciting project, Translational Approach to Improving Arrhythmia Mapping. So, Brian, welcome.
Thank you so much, Dr. Fedorov, for those kind words. You're gonna make me cry again. So first off, thank you to the student council for nominating me to give the talk during the medical student grand rounds. And I'll be presenting on my PhD research that I did through the medical scientist training program here at OSU. I'm currently in the third year of medicine. And so I've met some of you, part of my new class, and I'm excited to meet more of you. But this is what I've been doing for the past, like Vadim said, 8 years. Just to go over the general outline of my talk, I'll first be talking about just an introduction of cardiac electrophysiology and atrial fibrillation and what that means to mostly the first years who may not have had their cardio block yet. And then I'll go over some of the 3 projects that I completed. One is just identifying the mechanism of human atrial fibrillation. Then how can we validate clinical approaches to map that mechanism? And then finally, how can we improve upon these clinical approaches to treat human atrial fibrillation? First, a background of electrophysiology. So a myocyte, or a heart cell, really gets its activity, its signal to contract through an electrical signal. It starts as a resting cell at with a negative potential, and then its neighboring cell tells it like, hey, wake up, and it gets depolarized through sodium channels that increase the positive ions inside the cell. And then that really initiates the contractile mechanism. And then it cycles back down towards that resting negative potential. But during this repolarizing, it can't be activated again until finally it reaches that resting membrane potential, and it can again be activated in the next cycle. Heart cells don't activate independently, or they don't work independently. They are connected in like what's called a syncytium. That means the signal from one cell can spread to the next cell through what's called gap junctions. So the ions move through these pores between cells. And you can see from the cartoon here that there are more gap junctions along the long axis of the cell than on the short axis. And that really dictates how the speed of that electrical wave. So it goes faster in one direction than it does the other direction. But you can also imagine that if there's scar tissue or extracellular matrix called fibrosis, that may decouple myocardial cells and affect that natural wave of communication. And so if there's one cell activated, it can't go one way because the other cell is still in that repolarizing phase. It can't get activated. So instead it goes the other way. But then it also can rotate around this scar tissue, and that can cause abnormalities in the conduction system of the heart. Normally, that electrical signal starts in your sinoatrial node here in the right atrium. That will generate a normal rhythm at like 60 beats per minute in a normal healthy human. That then spreads to the rest of the atrium, and then hits the atrial ventricular node here in yellow, which then communicates the electrical signal down to the ventricles, where it then stops, and then the sinus node can start the next beat again. And that's what's called normal sinus rhythm. But in some instances, that normal sinus rhythm is disrupted, and we get what's called an arrhythmia. And this rhythm strip, this ECG rhythm strip, is showing us atrial fibrillation. For the med students, that's irregular R waves or ventricular beats, and a lack of clear P wave activation. Atrial fibrillation is the most prevalent sustained arrhythmia, and it's affecting currently about 7 million people in the United States, but that could go up to about 16 million with the aging population. And atrial fibrillation is so dangerous because it's the leading cause of stroke. Instead of the atria pumping the blood smoothly to the ventricles, the blood kind of swirls, and that can cause a clot, and that clot can then travel to the brain and cause a stroke. And so patients with atrial fibrillation are often taking blood thinners or anticoagulation drugs for the duration of their lives. Atrial fibrillation is also progressive disease. So comorbidities such as high blood pressure and obstructive sleep apnea and diabetes all cause a remodeling in the heart, which can predispose to atrial fibrillation. And then once episodes of atrial fibrillation start, they may only last a few minutes to a few hours at the first, but then every time they reoccur, they get longer and longer. And the longer you're in atrial fibrillation, the more likely a clot is to form and your risk of stroke increases. So it's more of a vicious cycle of progression of atrial fibrillation from episodic or paroxysmal to more persistent and permanent forms. Current treatments for atrial fibrillation include antiarrhythmic drugs. So you can have rate or rhythm control. These include your classic class 2 beta blockers, as well as class 3, like amiodarone, is kind of the mainstay of atrial fibrillation treatment. But as many of you know, antiarrhythmic drugs have a lot of unsafe side effects. And what's sometimes antiarrhythmic in the atria could be proarrhythmic in the ventricle. And eventually, due to that progressive remodeling of the heart, these can be rendered ineffective in the drugs. Another cure to treatment to atrial fibrillation is what's called electrical cardioversion. So the patient comes in the hospital, they're sedated, and electrical shock is delivered, which kind of resets the heart, and hopefully sinoatrial node takes over again into normal sinus rhythm. But the time between electrical shocks usually gets shorter and shorter with that disease progression until eventually it's no longer effective. Back in the 90s, they discovered that radiofrequency catheter ablation, or burning a small piece of tissue, can provide a cure for the arrhythmia if the tissue that you burn is the source of the arrhythmia maintenance. So currently, the standard of care for this ablation procedure is what's called a pulmonary vein isolation. So here we're looking at the back of the heart, left on the left, right on the right, and we can see the pulmonary veins coming into the left atrium. And so there have been implicated, the pulmonary veins have been implicated as the source or triggers that start the arrhythmia. And so you can burn or create a scar tissue that will electrically isolate the pulmonary veins from the rest of the tissue. So you're preventing that trigger from coming in. But with more sustained forms of atrial fibrillation, persistent, it's no longer dependent on what starts it, it's more dependent on what keeps it going. And so in long-term success of this pulmonary vein isolation is only about 28% in persistent AF patients. And so then, but you can consider this as a generic and an anatomy-based approach. So every patient has the same anatomy, pulmonary veins, and so every patient gets the same treatment, pulmonary vein isolation. But the maintenance, the mechanism of atrial fibrillation maintenance may be more patient-specific. So you may have to find where in the heart you have to ablate instead of just going off a generic procedure. And to do this, clinicians use an electrode mapping system. So you can consider, like, this is one form of electrode mapping. It's a basket catheter that's introduced through the femoral vein, travels up the inferior vena cava, and then is deployed or expanded like an umbrella inside the right atrium or left atrium. And then it records the electrical signal from 64 evenly spaced pieces of metal called electrodes. The electrodes can tell you when the atria is activating in that location, and then you can create maps. So you can see where the electrical signal is going and where it's coming from. And if you can identify a sustaining mechanism in that electrical signal, you can then ablate it. But unfortunately, people using the same technique for mapping get very different success rates. The inventors get about 80% success, and then other groups get back to that 25–40% success, as with pulmonary vein isolation. Furthermore, there's now a myriad of techniques and devices used to map atrial fibrillation on the market, each one purporting about 80% success, but when other groups use them, they don't get as much success. And they also see a wide variety of that sustaining mechanism. Each one says ablate here a different way. So here are some of those mechanisms that have been proposed from those clinical mapping systems. So one is what's called single reentry. So you can imagine the electrical wave like a car on a racetrack going around and around. It's now self-sustaining in a loop. It could also be like a hurricane, which is drifting around a single point. Another mechanism is what's called ectopic or a focal driver. You can imagine a group of cells which periodically will fire and drive the rest of the atria. Excuse me. That may look like a ripple moving on the surface of water. Another one can be multi-wavelets, and that's where there is no single location for a driver, but these waves can split into two can split into four, and then you can imagine if one wave gets extinguished, you still have three that can split into six. And so as long as the splitting of waves outnumbers the extinguishing of waves, you'll have a sustained arrhythmia. And then finally, you can have a combination or multiple reentries, in which case, if one reentry is terminated, another one may pop up in a kind of a whack-a-mole situation, which may again further complicate treatment for their mapping. This can all be considered complicated, the mapping of atrial arrhythmia mechanisms, due to the 3D complexity of the human heart. So the ventricle is known to be a thick muscular structure, and the atria has been considered a two-dimensional structure. But as you can see from this 3D reconstruction of a human atria from MRI, the atria cannot be considered as a simple two-dimensional surface, but has its own complexities within its wall, different wall thicknesses, different fiber orientations, and fibrotic or scar tissue within the wall itself. So that leads us to a classic example from one of Dr. Fedorov's favorite books, The Little Prince. And in it, they proposed, like, what is this drawing of? And you can think maybe like a hat or something. That's what I guessed. But if you look closer, or if you could see past the surface, it's really a python that ate an elephant. Not something you can tell from the first picture, but only if you can see below the surface could you really know what it is. And that's kind of the principle that we've been using to address atrial fibrillation mechanisms. You may have one mechanism within the depth or beneath the surface of the atria that, when viewed on the surface itself, could be seen as a totally different kind of mechanism, or even hidden within the structure of the heart. And that brings us to our main hypothesis, that atrial fibrillation in the human heart is driven by intramural or reentry within the wall of the heart, and that fibrillatory conduction can cause the surface activation to appear as breakthrough or even unstable chaotic waves. But if you can find the reentrant driver, which is truly driving the arrhythmia, if you ablate the reentrant track, then it will have an acute effect on the arrhythmia. It'll either slow it, organize it, or terminate it to sinus rhythm. And that's the cure for atrial fibrillation that we're looking for. Currently, in clinics, they use, like I said, electrodes to map the heart. And this cartoon shows kind of an example of what the limitations of those electrodes. You can see that the pieces of metal in blue have a very narrow field of view. They also have a very shallow depth of signal collection. So you can see that it might not collect signal from inside the atrial wall, and that they were missing information between the gaps between electrodes. To overcome this, there is a technique where you can use light to measure the electrical signal within the heart. There's molecules called near-infrared optical dyes, which are voltage sensitive. So they have a different color, whether the cell is activated or whether the cell is resting and polarized. And we can use that change in fluorescence to map how the electrical propagation moves through the heart. This system is more advanced than the electrodes because it can penetrate deeper within the tissue. And if you can imagine, if we can map from both the inside surface of the heart and the outside surface of the heart, we can now cover the entire thickness of the heart and not miss any electrical activity and really see the true mechanism of atrial fibrillation. Unfortunately, this light-based optical mapping system is currently only available to map the heart outside the chest. We can't get these lights inside the chest right now to map in patients. And so most of the studies that have used this mapping system have done so in mouse or pig or sheep hearts. Here at OSU, we have a more unique approach. We do this optical mapping system in explanted human hearts. So we get human hearts directly from patients who either undergo transplant at OSU. So when someone gets a new heart, we'll get their old one for study, or through the organ donation program, Lifeline of Ohio. When they have an organ donor who has consented to research, but their heart is not good enough for a new patient, we may get the heart instead. And through this program, we can now study directly the human heart, which is very different than an animal model. Over the last 8.5 years now, we have studied about 198 human hearts. Some of those hearts come from a car crash patient who has like 19 years old and has a very normal, structurally normal heart. And that can be compared to patients who are 60 years old with end-stage heart failure, which is a very remodeled and diseased heart. And so you can imagine some of these 198 had atrial fibrillation. And in those hearts, we can induce that atrial fibrillation again outside the body, and we can study it. Here's a movie of the heart beating outside the body. This is normal sinus rhythm, and we're able to revive the heart outside the body by using a perfusion system. So you can see tubes here running into the coronary arteries. We're pumping in a solution called Tyrode's solution, which has the same electrolytes, glucose, oxygen, and a buffer system as blood. It just doesn't have the cells or the proteins. And with this, as long as we can keep the pressure right, the pH right, and the temperature right, the heart is fooled into thinking it's back in the body, and it'll start beating again on its own. And then we can study how does the electrical propagation move during normal heart rhythms and abnormal heart rhythms. Importantly, this can now be done without any risk to the patient. So we can study experimental drugs. We can ablate regions of the heart we may not ablate in patients. And we can improve atrial fibrillation treatment. Here is the optical mapping system. It may look complicated, but it's really just a series of cameras and lights. The lights stimulate the optical dye, which then lights up either when it's activated and doesn't light up when it's inactivated. And then our special camera system can be used to tell the difference between those two with a filter. And we can then map the fluorescence as it moves across the heart. Here is a movie of optical mapping of a normal sinus rhythm. You can see blue is resting cells, red is then the activated cells. It's starting in the high right atrium, right to where the sinoatrial node would be, and then spreading uniformly across the right and then left atria, and this is normal sinus rhythm. From these movies, we can then create maps, and this is what I'll be showing for the rest of the presentation, where the electrical signal starts in the dark blue area, and you can see it moving smoothly to the dark red area. So we can see where it starts and then where it goes. And with this system, now we can start studying abnormal arrhythmias such as atrial fibrillation. And that brings me to our first project, my first project, where we're trying to identify the mechanism of atrial fibrillation by optical mapping in explanted human hearts. Here is an example of an explanted human right atrium. It's now been opened up. So it's been cut along the crista terminalis and opened up. And now this is the inside surface of the right atrium. You can see all the pectinent muscles here. When we pace the atria, the electrical propagation will move smoothly across the surface of the right atrium. And if we look very closely at this area in the black square, we can use a simultaneous camera system that's mapping from the inside surface, or the endocardium, and the outside surface of the epicardium. So from our endocardial camera, we get a very different pattern than from our simultaneous epicardial camera. And we wanted to explore what is the cause of the differences between these two activation patterns. And the underlying structure is the answer. So again, we can't consider the atrium as a two-dimensional structure. Its 3D architecture has to be considered. And we can study the 3D architecture of the human atrium using what's called contrast-enhanced MRI. The contrast we use is gadolinium. It's a heavy metal that diffuses into the heart tissue, and it hangs out a little bit longer in scar tissue or fibrosis than it does in normal myocardium. So when we scan our tissue a few minutes after we inject the gadolinium, we'll see a brighter signal from scar tissue and a darker signal from normal myocardium, and we can see where that scar tissue is. So when we go back to that example of differences between the endocardial surface and the epicardial surface, we can then quantify that difference with by just subtracting the activation times, and we can see some areas are very similar in activation times, while others in this dark red area you can see is very different in activation times. And so by looking at the structure, we can see why. So here's a cross section from the outside surface on the top, inside surface on the bottom. Now we're looking at the inside of the heart tissue. We can see where we paced. There's a lot of white pixels, and this is all that scar tissue. And then the biggest difference between the epicardial and endocardial surface you can see is caused by the thickest tissue. You can also imagine that electrical signal can't jump from number 2 up here to number 3 over here on the endocardial surface, but it can run smoothly between 2 and 3 on the epicardial surface. And that's really what we see the difference in our activation maps. That difference can also be explained by fiber orientation. So if we're activating the heart from top to bottom, it'd go faster along the epicardial surface, which has blue fibers going up and down, than the endocardial surface, which has red fibers, which are showing right to left orientation. And so in general, we saw in every heart, there was some difference between the endocardial activation and the epicardial activation. So there's really a difference between the surfaces of the heart. And that difference gets more pronounced the faster we paced. So you can imagine during a very, very fast atrial rhythm, like atrial fibrillation, that difference between the endocardial surface and epicardial surface could almost be thought as 2 different layers. So here is an optical movie showing the endocardial surface during atrial fibrillation. The untrained eye can look very chaotic. And so we'll look and see what the epicardial surface shows us. It's different, but just as chaotic. And I can tell you only because I've stared at these movies for hours on end, that if we look at the red box here on the endocardial surface, we can see a reentrant pattern or one of those self-sustaining mechanisms of atrial fibrillation. If we then turn these into those maps I showed before, we can see on the endocardium, it's going smoothly from dark blue all the way to green to yellow to dark red back to dark blue. But on the epicardial surface, we get a breakthrough pattern. So it goes dark blue and spreads out, but it never really comes back to that origin. In general, we saw that 17 out of 20 atrial fibrillation drivers were seen as reentrant on the endocardial surface, while simultaneously only 8 were seen as reentrant on the epicardial surface. And this makes sense, again, if we think about the structure of the heart. Here are the pectinent muscles I showed before, and we saw in this heart that the reentry is running along very specific sets of pectinent muscles, which are an endocardial structure, and why they may not show up on the epicardial surface. And this is important because if you were only mapping from the epicardium, you can see that translates to this red star here on the endocardial surface. So if you ablated this region only, you may entirely miss the reentry circuit or the sustaining mechanism. So we really need to know exactly where the sustaining mechanism or reentry in the heart is to plan effective ablation. With that said, with our optical mapping system, we're able to test different ablation strategies without any harm to patients. And we found that what's called what we coined the term smart ablation, or a substrate modifying ablation of reentrant tracts, is the most efficient manner to terminate reentrant arrhythmias. So first, the reentrant driver is found, and then by ablating the reentrant driver, we're able to organize the arrhythmia into a slower. But you can still see every time we ablate, we create more scar tissue for the electrical signal to rotate around. So we must connect the original ablation to an anatomic obstacle. In this case, it's the tricuspid valve. And once that's connected, then we can terminate the arrhythmia and prevent its reinduction, and hopefully provide a stable long-term cure for atrial fibrillation. So in conclusion for this project, human atrial fibrillation is driven by intramural reentry that can appear as focal sources if viewed from the surface only. Intramural reentry reconciles the diverse mapping results seen clinically. So in some instances, if you're only mapping from the surface, you may not be able to see the reentry, and instead you see multi-wavelets, focal sources, but really underneath the tissue, just below the surface, is that intramural reentry. But now that we've identified that mechanism in human hearts, how can we translate that into clinical practice? So to do that, we took one of the clinically used mapping systems, that basket catheter I showed earlier, and saw how does it compare to our optical mapping in the ex-vivo heart. Again, clinical studies that have ablated atrial fibrillation drivers with this system show either very good success or very bad success. And we have to kind of reconcile, how does the same technique have such different success? And it really comes down to subjective nature of mapping. So this is the map that you can acquire from a basket catheter. And this is what a clinician has to interpret during the mapping procedure in order to know where to ablate. So kind of like before, we're looking for a self-sustaining either reentrant or focal mechanism. And just staring at this, I wouldn't be able to tell you. And there's such a steep learning curve, and that was cited in this study that showed low success. It's really up to the clinician to decide where to burn someone's heart. But again, we can integrate that in our ex vivo situation. We can combine that with our optical mapping, which shows very high resolution and subsurface signals. And then we can ablate without any harm to patients, and then correlate that to the underlying structure with MRI. So now we've integrated optical mapping, electrode mapping, and MRI on the same human heart. So first we compared how does normal activation look by optical mapping compared to electrode mapping. These two maps on the left shows optical mapping going smoothly from dark blue in the high red atrium down to red in the left atrium, and that's very normal. And that's copied by the electrode mapping here, again showing dark blue to dark red. But that's a very slow, even rhythm, and not the fast, chaotic atrial fibrillation. So first, to compare how does electrode mapping stand up to optical mapping during atrial fibrillation, we looked at the frequency recorded by each modality. So that's how fast is the rhythm going. And we can see that the speed of the rhythm is not uniform across the atria. There's some areas that are faster and some areas that are slower. But importantly, the speed shown by optical mapping on the left correlated to the frequency seen by electrode mapping on the right. And that was shown to correlate across all the hearts that we studied. So, so we can show you where is there fast and slow activation, but now we have to know, does it really show us where that reentrant driver is so we can plan our ablation? Here's another one of those movies now in our ex vivo setting of the electrode mapping. And again, it could be very difficult to interpret these without expert or a lot of experience. But with our ex vivo optical mapping, we can now zoom in on one of these areas, and we can see in the top right corner, there is indeed a reentrant driver underlying the chaotic signal by the electrode mapping. Importantly, at least this electrode mapping system doesn't make you interpret all these maps on your own, and instead provides a software algorithm that can analyze these signals and show you where it thinks reentrant drivers may exist. And in this case, you can see it had highlighted this area in the top right corner, which matched the driver visualized by optical mapping or validated by optical mapping. And in general, this algorithm, which is called FIRM, correctly identified 79% of AF drivers. That means it missed 20%. And then there's another phenomena here that I'd like to point out to everyone, is that there's areas outside the driver that are highlighted by the system itself. And so what's happening out here that's causing the system to think that there could be drivers there? Here's another example, more extreme. In the previous example, there's only one red spot, and so you could probably guess that you should ablate at the one red spot. But in this one, we have 5 spots, all equally red. And so the clinician has to decide from these, which one do you start with, which one do you ablate, do you ignore any, or do you ablate them all? And by combining with optical mapping, we were able to show that only the 2 spots on the right were actually related to a reentrant driver, while the 3 on the left were false positive annotation that were caused by what we call passive rotation. So the electrical wave was coming from the right off screen here. You can see the blue, and then it came and it turned and then stopped. But because it always came, turned, and then stopped repetitively, it fooled the system into thinking it was spinning in one point in a self-sustaining manner. But really the waves were always coming from the driver on the right. Importantly, when we ablated the point here, we are able to terminate atrial fibrillation and prevent its reinduction. So if you think of a clinician trying to interpret this map, if they ablate all 5 points, they will hit the reentrant driver, but they'll also create new scar tissue that could be pro-arrhythmic in the future And that can cause a lot of the discrepancy we saw in the results between studies. So people who may have more familiarity with the system may know to ignore these sections, but other people, if you just sell your system to everyone, they may not have the familiarity to focus only on the driver region itself. And so we try to find a way that we could distinguish what we call true drivers from false positives. And unfortunately, we are unable to figure out a way to distinguish them based on these electrode mapping systems themselves, because they're all very similar. As you can see, they're all bright red. The activation patterns were all just as chaotic. So we had to go to a different modality. And again, we turned to the underlying structure of the heart to provide an answer. So we used, again, that contrast enhanced MRI. It can show us where fibrotic tissue is. Here is the raw MRI in the top left. And then if we color code where the fibrotic tissue is, we get the top right image here. But you can see that even by histology of the same section, so this is the gold standard fibrosis mapping, but again, can only be done outside the body. We see that there's a thick border of atrial fibrosis on the endocardial inside surface of the heart, and that there's also some fibrosis on the outside border of the heart, and then there's some in between the muscle, but it's scattered throughout. And then you can imagine that if you have dense connective tissue only on the outside or inside of the heart, it doesn't really affect the electrical signal between cells. And so we came with a hypothesis that what if we only look at what we call intramural fibrosis? So that's only the green and yellow fibrosis here between, that would occur between the muscle cells themselves that could be arrhythmogenic. And we mapped that to their example previously. We saw that indeed this intramural fibrosis was located more in the driver region than in the false positive regions. And we quantified that across all of our hearts. And we showed that intramural fibrosis is higher in driver regions than non-driver regions, and importantly, false positive driver regions. This wasn't true for subepicardial or subendocardial fibrosis. And importantly, it was not true for total fibrosis, which has been looked at in previous studies. And they also were unable to show any correlation between drivers and total fibrosis. But we suggest by using only intramural arrhythmogenic fibrosis, can you distinguish between drivers and false positives? So in conclusion for that project, validation of multi-electrode mapping is required to evaluate what a true and false positive AF driver looks like by clinical electrode mapping. And importantly, integrating multiple mapping modalities can increase the sensitivity and specificity of AF driver identification. So you can consider at least that there would be a functional mapping done by electrodes in the heart that could show you more than one, that could show you multiple drivers. You can then compare that against an MRI that the patient got previously. It can show you where the scar tissue in the heart exists. And then by combining those two modalities, it can show you with higher sensitivity and specificity where that driver is, or at least where you should start your ablation protocol, because some of these false positive drivers may be ignored. And then finally, we should, for the final project, we validated previous mapping, clinical mapping, that basket mapping approach. But now we wanted to see how can we improve that in patients. So up until now, I've been kind of presenting the Reentrant AF driver as a single reentry circuit, so spinning around a single piece of scar tissue in the heart. But of course, the heart doesn't have one piece of scar tissue. It can have a bunch, and it has a network of muscle cells between all the scar tissue. So in reality, a Reentrant AF driver could be the combination of a bunch of different circuits all happening in succession. So you can think it goes one way, one time, and then the next time it can go a totally different way. And this can fool algorithms and also can fool visual inspection of maps. And it can make it a lot harder to find where these drivers are because the activation pattern isn't consistent. From our previous experiments in canine and human hearts, we showed that a molecule adenosine, some of you may be familiar as its properties for terminating supraventricular tachycardias. So anything that has a reentrant circuit through the AV node can be terminated by adenosine. But atrial fibrillation, its reentrant circuit is entirely in the atrium. And so adenosine actually speeds up the atrial fibrillation. And it can do that by stabilizing the reentrant circuit across a smaller, more condensed path. And so we propose to use adenosine to stabilize the multiple reentry circuits into a single common path that can then be easily identified and ablated. We first did that in our ex-vivo experiments. So we had a human heart induced into atrial fibrillation. We then perfused adenosine into it, and we can see the reentrant driver became much more stable and easily visualized. And importantly, the same phenomenon was visualized by the electrode mapping system. So by the low resolution clinical mapping system of this same episode, we saw that the AF driver went from a unstable focal pattern to a more stable reentrant pattern that could guide ablation more accurately. And so now that we could show that adenosine does help electrode mapping, and we can do electrode mapping in patients, our next logical step was, let's use adenosine for electrode mapping in patients. And so we were able to recruit 10 patients here at OSU with the help of Dr. John Hummel, one of our clinical electrophysiologists at the Ross Heart Hospital. And he uses that basket catheter technique to identify and ablate AF drivers. But sometimes it's harder for him, just like everyone else, to interpret those maps. And so he injected adenosine into these patients. So you start your map, you get a baseline recording of the atrial fibrillation. You then inject adenosine into the heart, and you can see it slows atrioventricular conduction and even can cause AV block for about 17 seconds, which is safe in the cath lab. That will then change how the pattern of atrial fibrillation is seen and could reveal a driver. Importantly, all of these patients had at least one driver that was ablated and successfully terminated the arrhythmia. So we knew where to look retrospectively. Here's an example. First, we have on the left the baseline AF pattern. And again, as been evident throughout the presentation, these can be difficult to interpret. But when we added adenosine into this heart, into this patient, we now see a more uniform activation. So that white activation is now going in a more uniform circle. And importantly, if you look back at the baseline activation pattern, now that we know where that driver could be, we do see some predilection of the electrical signal of going in that reentrant pattern. It's just different every time and harder to identify. But now that we know where the driver is, because we use the adenosine to clarify its location, we are able to ablate during these baseline conditions, because again, adenosine only lasts about 17 seconds, and successfully terminated the arrhythmia in this patient. And they had long-term success at 12 months. Across the 10 patients, we saw that their atrial fibrillation drivers had some different responses to adenosine. We saw that some got better, some were better visualized with adenosine, while others were better visualized at baseline. By separating those groups, we could see that indeed there was a group that significantly improved visualization with the injection of adenosine, but there was another group that significantly decreased. So in this group here, adenosine actually obscured the driver and made it harder to see. But if you can see, there's a kind of a clear cutoff between who can be helped by adenosine and who can be hurt. So it was more likely that if you had very low visualization of your driver at baseline, that adenosine was going to help you. So what we concluded from the study was that first map the atrial fibrillation at baseline, and if you can see a driver, ablate it. That's great. But if you're struggling, like is common in many patients, adenosine offers a tool that can be used to clarify those unclear maps and eventually improve patient treatment. So in conclusion of this project, reentrant AF drivers may travel along several paths within arrhythmogenic hubs, which reduces their perceived stability. Adenosine is a useful clinical tool to unmask hidden reentrant driver hubs in persistent AF patients during multiple electrode mapping. And finally, across all of the projects, our theory of intramural microanatomic reentry provides the first universal mechanism to explain all clinical mapping results. And reentrant AF drivers represent a critical target for patient-specific ablation and a possible cure for AF. And finally, identification of reentrant AF drivers in patients can be improved through integrated mapping and pharmacologic challenge. So in the future, we hope we can better identify and better treat atrial fibrillation drivers. And with that, I would like again to thank my mentor, Dr. Vadim Fedorov, as well as all of our lab members throughout the years. And this technique of optical mapping of the human heart is very challenging. So we get these hearts at any hour of the night, just like a transplant cardiac surgeon would. We also want to test as many hypotheses on each heart as we can. So each experiment usually takes about 18 to 20 hours. We then process all of the data in a retrospective manner, which takes a lot of time. So I'd like to thank all the undergraduate members of the lab who have done a lot of the mapping results, as well as our collaborators in the University of Auckland, New Zealand, who did all of the structural reconstructions of our MRI. And of course, I'd like to thank our local collaborators, Dr. John Hummel and Raul Weiss, our clinical EP specialists who helped us with the clinical trial, Dr. Orlando Simonetti, who helped us do MRI of our patients, as well as our clinical cardiac transplant team, Nahush Mokadam and Brian Whitson, as well as I would also like to thank our sponsors and our funding from NIH and the local philanthropists. And Vadim would hate me if I didn't say that there are student positions available in our lab. Please contact me if you have any questions about these projects. It'd be great. You can ask now a third year Alex Oreguin, who did a summer in our lab as part of his M1 to M2 summer project. I forget what that's called. But just email me if you have any questions or are interested. Thank you. And now I'd invite Dr. Fedorov back to moderate any of the questions.
Thank you so much, Brian. It was excellent as always. And I think, guys, if you saw the last slide of Brian Hansen, you'll see also the word honey badger. The honey badger, it's most fearless animal in the world. Yeah, thank you, Brian. You can read that. We have a name, our mascot, honey badger. To do this research, what Brian mentioned, you have to be on call 24/7. You have to just follow our cardiac thoracic surgeons, waiting for the heart, bring it to the lab and study for many hours. It's literally 24 hours experiment. So you have to be fearless, unstoppable, and never give up. And this is what Brian represents. This is what he's represent and represent the Ohio State University, that we never give up and taking criticism. It's important. So Brian, together we published 30 papers. It's not easy, and most like an easy paper was actually the last paper that he presented about our clinical translational application. It was reviewed 8 times. 8 times we fight with reviewers and eventually we won. That's because honey badgers never give up. So it's important, guys, for all your future career, and that's my advice to all students all the time. So when you're receiving the criticism for your fellowships, application, grants, papers, you have to always think how you can do it better. So, and pushing forward science and taking this criticism as advice rather than obstacles. So here I would like to take, guys, any questions. It would be great if anyone provide any comments. So I can read from Christina. Brian, can you please talk about the project? What wasn't going as planned and how got you through, either individual or with help of our team?
Oh, yeah, thank you. So like all experiments, we start with hypothesis, but it's not always so direct. So in the first project, we used a limited amount of hearts, and we saw that we had reentrant drivers that were very easy to see. But the more and more hearts we got, we saw that there could be patients with different comorbidities or different patient populations, that the driver isn't so perfectly seen all the time. And that's really what we had to take a step back and reevaluate our hypothesis. So were we completely wrong about these reentrant drivers? And we had to reanalyze our research and look and like, no, it could be that it's not the same reentrant path every single time. And that's what led us to think of it not as a perfect race car track reentry circuit, but more as a hub. So an area in the heart that is more prone to causing these reentry circuits, but it's never the same reentry circuit.
Thank you, Brian. So can you also mention about other cardiac arrhythmias which we studied together? So what else our group, our particular lab, as well as whole human heart group is approaching and studying?
Yeah. So specifically in our lab, we focus on atrial arrhythmias. So that includes atrial fibrillation, atrial flutter, atrial tachycardia, but also that natural pacemaker of the heart, the sinoatrial node. So that can cause bradycardia or the need for an electrical pacemaker implant when the sinoatrial node goes wrong. And so with this explanted human and this optical mapping method, we can look through the surface of the heart and see the sinoatrial node function in an isolated manner away from the atria, which can't be done in patients. And so we're also trying to improve and see if we can cure the sinus node instead of just implanting a pacemaker for the rest of the patient's life. But you can consider that we get the atria for study, but there are a lot of different labs, about I think 17 different labs at OSU, who each get a different part of the heart. So they're looking either, I know Dr. Moller's lab is looking at the ventricle and long QT syndrome, as well as atrial fibrillation. And then you can also do molecular studies and see which patients and what predisposing factors. So like diabetes may present with a different kind of heart disease than high blood pressure, or the combination of the two may have also a different synergistic effect that can be studied in the human heart now.
Oh, thank you, Brian. So I have another question from panel regarding, do we have any plan to do in vivo optical mapping in patients? Is something we're planning to do?
So that's a great question.
In vivo optical mapping.
Yes, so we have started that project, but of course there's a lot of steps between having something work in the lab and having something work in a patient. And one of those steps is animal research. And so we've started a large animal model of in vivo optical mapping. So we're able to use our camera and light system during open chest surgery. This is important for patients because a lot of patients with valve disease will have atrial fibrillation. And then they'll sometimes get their valve replaced during an open chest procedure. So that gives us the opportunity to translate our optical mapping into patients. But currently we're still doing those preclinical animal model studies. But a good question, thank you.
Thank you, guys. I have a question regarding, is any other option except cardiac ablation, which we know it's permanent damage of the tissue? And if we have this arrhythmogenic fibrosis hub, do we have any plan how we can treat that in other way, rather than damaging this, destroying this tissue forever?
So how I presented in my, the project 3, is that these reentrant drivers are moving around scar tissue within the heart. So something caused that scar tissue to form in the heart, whether it was high blood pressure, diabetes, age. So atrial fibrillation is very common in the elderly. So is there anything we can do, one, to prevent the formation of that scar tissue? Or once the scar tissue forms, is there anything we can do to remodel the heart back to a less fibrotic phenotype? And studies have been shown that weight loss is very helpful in that setting. It can improve the success rate of ablation. because it can prevent more remodeling of the heart.
Thank you, Brian. Brian, can you provide some advice to medical students and everyone who is planning pre-med undergraduates who is planning to be medical students? How is important, in your opinion, all this research, all this year which you spent on our lab and sacrifice? I know you sacrifice a lot from your personal life, and we know that no success without sacrifice. That's what Honey Badger do. But advice, how it's important, this research in the lab, what you've done and what you still continue doing. Thank you for that. For your future clinical career, would it make you better doctor? Would you do something more than if you just will be working as a medical doctor?
Yeah, great. Thank you so much. So from a personal standpoint, I chose to pursue the MD PhD route because I couldn't see myself not asking questions during my clinical practice. I wanted to have the training to be able to see patterns or recognize patterns within my patient population, and then thinking, is there a better way that we could do this? And then I wanted to have the skill set to test that, to implement a better way to do it. You don't need to have MD PhD training for that. Some of the best clinical researchers are MDs, but in their training, somewhere in their training, they decided to do research, and it really gives you a good opportunity to ask questions. So understanding the mechanism of disease can help you understand how we treat it currently, but it can also help you try to think of a better way to treat it in the future, especially with new technology coming out. So I know the hot one right now is machine learning. So this new technique has application across medicine, but it's going to take clinicians who have that patient population in order to implement it in a successful manner. And I think it could change the face of medicine.
Thank you, Brian. But how about advice to medical students? Do they need to do research during summer, during semester in the labs? Or all they
I think it's important.
Only?
I think research is important to all medical education because it shows you how much we don't know. So as a doctor, there's so much knowledge and there's so much we can study from the textbook, but understanding that there is unknowns will help you make more informed clinical decisions, especially for the patient in front of you who isn't a vignette. They are an individual with differences and unique features, which you can then tailor your treatment to that patient specifically. But research can help you understand what's unknown and better treat that.
Thank you, Brian.
Thank you.
And as I mentioned, guys, the Brian is represent the student successful example of most successful example of our Ohio State University students who actually will be a leader in the field and truly believe in that. And this is the guy whom I can believe if I will have any problem with my heart rate, that's a guy who's gonna treat me in the future. Thank you so much, Brian. So I'm very proud of you.
Thank you, Dr. Fedorov.
Awesome. Well, thank you, Brian, for that wonderful educational experience. I know I definitely took a lot out from this presentation and learned quite a bit. I want to thank all of our viewers who tuned in and those that will tune in on YouTube for joining us today. And just a reminder that our next presentation will be on the 1st of December, where Shan Lansing will be talking about her role in rural healthcare advocacy. So hopefully that you all can tune into that one too. And we thank you all for further supporting medical student grant rounds.
And remember to nominate your friends.